this? Okay. Okay, so welcome back everyone to for another mass seminar. So this week we have our very first external speaker, Hille from Leiden University, who is visiting Alice, and he will talk to us about multi-scale model-based investigation into the molecular processes that drive long-range oxygen transport in some plants.
Yeah, thank you. Mouthful. And thanks for giving the opportunity to speak here in Lancaster. It's my first visit here, and so far I enjoy it very much. And maybe we'll first start with a brief personal introduction that so that you can place a bit the research that I will be talking about. So I'm working at the Math Institute at Leiden University in the Netherlands, and actually I have two sides. One is more fundamental mathematics and topics like dynamical systems and spaces of measures, and I like to also mix deterministic probabilistic systems like piecewise deterministic Markov processes. Another topic I'm working on a lot with Alice here is model approximation, all geared towards actually applications in towards biology. So other major work I'm doing on mathematical biology, and really I like to collaborate a lot with experimental plant biologists in Leiden, in what we call the plant biodynamics lab there. Main contributors to the talk that I will be giving are the people here from the Institute of Biology there. What I will be talking about is joint work between the math department, my side, and the plant biodynamics lab, and it's about this simple weed that you may find in the street. It's a model system in plant biology, the E. coli of plant biology, the Arabidopsis thaliana it's called, and it's about long-range polar oxygen transport. And I will introduce you later to the biology, so that you have a bit of an idea what it is about. The next step is then that I want to discuss a bit how we approach the modeling of this phenomenon, this transport in this weed, and also how we really integrated experiment and the modeling work, and we found some interesting things I think that are worth sharing. But the main part in the end, I want to focus on actually how we use let's say the ultimate model that we developed to explore and to understand what actually happens in this polar oxygen transport in the living plant, and various issues that we had to overcome to be able to draw conclusions. So first the introduction, more to give you an idea of what this plant is. It's a model system because it's pretty well known. It's fully sequenced. It's you can breed it quickly, and what is important for us is various mutant plants available, such that we can link the experimental modifications to the physiology of this plant, and the modeling work on the other side to gain insight on what happens internally. The oxygen transport was in the title, and oxygen, that is a plant hormone. Plants have also hormones like animals to communicate particular states of the overall system to other parts of the system. Oxygen is a kind of tiny molecule you see here in the chemical structure. It's highly important in various processes in growth and development, controlling cell division. It determines patterning when side branches pop out, for instance. And for that reason, also in the plant growing industry, which is quite a business in the Netherlands, one is interested in how to control side branches, how many fruits there are, etc., especially for crop plant. So that's why we want to study this transport, and in particular the long-range transport from the top of the where auxin is made to the roots. And that's called polar auxin transport because it's directed top down. The motivation for studying this is really because it's important plant hormone. So in fundamental biology, one is interested in how it works. But what I mentioned too is that it has some interest in the breeding and to get more fruits on plants, like tomatoes, to be able to control. And there's also another interest that already in the mid-1970s, a theory about this was formulated by people Rugery and Sheldrake, Raven, and called the chemiosmotic theory. I come back to that slightly later. But still, up till now, or just recently, there was no quantitative, really true experimental validation of this theory. And there was also questions about where this transport was taking place. So there were quite some questions open that could be addressed. And on the other hand, if you consider this topic, there's really good opportunities for modeling, model-based analysis, because this auxin molecule that is very small, but there's some delay now. Yeah, there it is. The molecule is very small, so you cannot attach a fluorescent protein, for instance, to this small molecule and make it visible in the plant, because that would be something like attaching a huge ship to a small sailing boat, and then all properties of the original molecule are gone. And on the other hand, you can use radioactively labeled auxin, tritium labeled auxin, but then you cannot have a well-resolved picture in time. You can see very accurately how much there is in a particular part of space, but then you need to cut down your part of your plant, so you can do that only once. But then with modeling, you can get more insight. And for the modeling, the first question was, where does this auxin actually is in the plant? So this going top down, that was already postulated by Darwin in his time. So it's pretty long. In the 1970s, we have then a theory how it was transported. But if you cut a stem and you have a cross section like this, using so this is the biological side. Special promoters that act on auxin, you can make it visible in green where most of the auxin is, and that is in these parts of the stem section, which are called the vascular bundles, that are these pie-shaped parts recognizable in the plot. So clearly there you should look for the transport. How it works, and I will briefly get through it because it is important for understanding how the model came in later, is this chemiosmotic theory. And the theory says that, well, in these vascular bundles, in the vertical direction, you have cell files, and the auxin hops from one cell to the other. And in between the cells, because it is a weak acid, it will split according to an anion form and a protonated form, depending on the acidity. And in between the cells, it is acidic, and inside it's more or less neutral, making that inside the cell, 99% is in anion form, and here it is about only 40. And then it moves from the intermediate space, but because the cell wall, the cell membrane is charged, the anion cannot easily get through. The protonated form can, but there are inside, it is the opposite. The anion form cannot get out, so you get an ion trap kind of mechanism. If you put then the molecular biology on top, then you have carriers here that facilitate the transport. So you have actually two parts that are important. You have one that is from the red family of aux-lax proteins that pump oxygen into the cell, and mainly essentially in the protonated form, you could say. And on the other end of the cell, there are the green ones, and these are from a family of PIN genes, and they are famous for being recognized as oxygen transporters, and they pump out the anion form. And when you look at how they are distributed around the cell, so the red ones, the pumps into the cell, are even more or less homogeneously distributed around the cell, and the PIN ones are located at the bottom line, so from the top to bottom, pumping the oxygen down. That is the polar direction. And in modeling this whole process, you need to bridge scales in this. So on this side, we have 100 micrometer long cells, but also need to consider the microbiology of the transport through the cell membrane, which is a few nanometers, of course, 10 nanometers. Then these files, they sit in these vector bundles, and yeah, they're part of the cross section of, say, 2 millimeters diameter. But then the whole inflorescence stem is much longer, so the polar transport, and we did measurement on 60 millimeters long stem segment. So you have a huge spatial scale. And yeah, you need to overcome that from a modeling perspective and an analysis and statistical perspective. So that is where I will focus on in the talk. Slightly more biology or measurement to see how to appreciate what data you can get from this, and then to draw conclusions. So what we did is take a 16 millimeter long stem segment and you cut it out, and then you can place it in this petri dish filled with paraffin wax, and then you have a donor well and an individual receiver well. You seal them into these bridges with wax, and then you can fill the donor well with radioactively labeled oxygen, and then you can see how much is transported through the stem segment by regularly sampling the receiver well, and then you get this kind of curves, transport curves, that you see how much gets transported through the individual stems. And here, this is a typical, yeah, you see the biological variation in their transport behavior among different plants of the same genotype. But what you also can do at the end of the experiment, you take out the stem segment and cut it in pieces, and then measure the content, the oxygen content of each of the pieces. So then you get what we call a tissue profile, like this. If you put the stem segment in the proper order from top left to bottom right, you will see this dark green profile. If you reverse it, then oxygen cannot get through, and you see a kind of more diffuse it. But it's a type of data that you can get, and there's not much more to get, not more detail. So how to draw then conclusions and on what happens on this microscopic scale, where we have these individual proteins on the membrane of various types? Well, we can only measure here at this top microscopic scale, and here at the microscopic scale we can make interference by considering mutant plants in which, yeah, various of these proteins are present, dysfunctional. You can make variations. So we need to get a model of what I now want to discuss is a bit of quickly show you through how we got to a kind of say ultimate model. It's not the best model in the end, but the ones that we're satisfied with, because there are some nice things about it that can be of interest, I think. And when we first started this, we were thinking, okay, how to proceed, how to start with a model. Now, on the one hand, you can think of available knowledge about mechanisms in the system that you can have from low to more, many, and on the other hand, you can have lots of data available, and the other hand, hardly any data that you need or is available. And you can think of different techniques. So let's say the machine learning approaches, you need lots of training data, but not so much specific knowledge. You can use that to gain insight in what could be there. On the other end of the spectrum, you have the mechanistic models where you need a lot of knowledge of the physics and how everything interacts. And then even you can draw up models with hardly any data. But in our case, we have in the middle, the phenomenological modeling approach, where we have a mix of both, because we don't know whether the chemiosmotic theory is right. We want to test that, and then we want to gain more insight. So we in this middle section, and what we did was starting from the existing models from the 1980s. And that is by Mitchison, and he started with this already modeling the chemiosmotic theory, but with a cell file, like here, equals cells. And inside the cells, you have simple diffusion, transport by simple diffusion. And then from cell to cell, you have a flux. And the flux from the I to the I+1 cell is depending on the concentrations at the ends in either cell. And there are two parts in this. There's a diffusive symmetric part, which parameter Q that will be relevant here. That's the strength of the diffusive aspect of the transport. And there is the asymmetric part, which is the polar part with the parameter P, and the P determines the strength of that. And the stronger P, the stronger the throughput is in the end. If you make this into, you actually have a whole system of coupled diffusion equations. In around 2000, Eric Kramer started approximating this by a single advection diffusion equation. That's not so, let's say, new, but he was able to compute the velocity, the effective velocity and the effective diffusion in this microscopic view of this transport in terms of the underlying parameters. So that's kind of upscaling part, and this is an example of model approximation that we are also, Alice and I are also interested in other cases, that you have a detailed model and an effective model that want to link the parameters at different levels. So this one will reoccur later. I will abbreviate this to the MK model for Mitchison-Kramer. That was our starting point, and we started building our model. So we have this experimental setup, and we then needed to actually model this setup. So at the core is the Mitchison-Kramer model, and we have to expand with the loading and unloading dynamics of at both ends. And that is done with dynamic flux boundary conditions. So you have a pool with an ordinary differential equation for the concentration of labeled oxygen in one end, and an influx here. And the influx, that was a bit tricky at the part how to include it. And in the end, it turned out simple. You have in the advection diffusion equation, you have the boundary condition that you usually have on one side, the diffusion and the advection. And what you say is that the diffusion advection at this end is precisely what can go through the barrier here, and the flux through the barrier is a kind of diffusion type of description with how much can get in from this donor well and how much can get in the opposite direction out of the stems, leak out of the stem segment. And that you can do on both sides. And then on the other side, you also have an ODE. So that is the system of mixed type where the boundary conditions are dynamic. Right. If you now start that and look at the data that we had. So in blue, you see the measurements of what happened, arrived in the receiver well. Then we were first very content. Okay, this is a nearly perfect fit. We can fit this, we're done. Then, yeah, we thought, okay, let's try some new experiments, cut up the stem segment in parts and see how much is in there at the end of the experiment. And then we got this. Was the blue are the measurements and the red the simulation, and that was completely off. So there's something missing. And yeah, so the model was dismissed, and we expanded first to include some loading in the tissue surrounding the vascular bundles that improved a bit the situation, but not fully. So that was also not good. You try other things that you start discussing. Okay, maybe some of the oxygen gets immobilized. So we had the version with actually a distributed ODE in there. So oxygen gets fixed. And another one is, okay, it may also leak out of the system, and you get another alternative. And yeah, you start like leaking from the transport file to the neighboring cells. That is, and the U is the central component, the Z is the immobilization, and the W is the what leaks out.
I just asked.
Yeah, of course.
Without the cutting, the first thing you showed us seemed to match experimental data so well. So if you don't cut it,
if you don't cut, you would have, oh, this is nice, this is done. But yeah, at some point it's good to try play around, and that's it's mainly their professional expertise that they know. Oh, let's try this or try that, and then suddenly we found something new that we okay, hey, this is not good.
But is this cutting somehow reminiscent of the biological phenomena you tried to model?
No, but it had been used before that people did experiments and then cut up the stem segment and measured. So it was an idea, okay, maybe try this as well and check whether we see the same thing. So that didn't came fully out of the blue. We had then these two, and then they invented a new experiment that halfway in the experiment they replaced the radioactive donor by a neutral donor. And then you are watching how the stem segment will empty in time. So that is what you see here. After this point, it will empty. And now the immobilization model could follow that perfectly. Well, if you looked at the leakage model, then this is blue is the data with interpolated data, then the model didn't follow it. Wasn't able. So this one was, and also at the end of this experiment, it was completely off in the tissue profile. So this one was need to go out. So you're left with the top one. Now that idea was okay. Bring in the mutants. So one of the mutant plants is where all these import transporters, the red ones, are knocked out. And the plant looks completely normal, but you get a different kind, more of a diffusive type of profile in the tissue. And yeah, one saw that again that this model could fit that also well. And you think, okay, we're done. No, because what you had good fits, but what turned out that there was a change in parameter values between wild type and the mutant, and the change, the direction of change in these parameters could not be explained biologically. So this was not an explanation of, it was a good fit mathematically, but there's no logical explanation of what happened. So that was also not good. And then you get desperate, and so we tried to put them together and see what happens. And that in the end worked well, and I will show how that worked well by it's with Hille now. It's a difference. Yeah, there it is. So this is a combination of the two, and yeah, you get to a complexity that is requested more or less by the data. So it is, yeah. And get to what we did with the model. So if you then look at the model, you can start looking inside and see what happens. So you're looking inside now, touch a stem segment and see a pulse traveling through. And we started looking in the literature, and we were happy to find that already in 1967, Mary Goldsmith cut up various stem segments at different times and found similar type of profile behavior. And so, okay, that's confirmed that this was fine. We also started doing a parameter sensitivity and identifiability analysis. Some are difficult to measure, but others are can be estimated well. So the parameter v, that is the effective velocity, that can be very well estimated. The system is very sensitive to changes in v, in quality of fit, and that is visible here. So you see percentage. This is the total cost function that is optimized, and only a relatively small change in so up to 3% accuracy you can get on the v. So it was. What we also found, and that was by trial and error, that some parameters were interchangeable. And usually you find that by doing non-dimensionalization of the model, and then you get quite a lot of information. But for these two parameters, it's not so important what they precisely are, but it was not apparent from non-dimensionalization, but by playing with the model, we found that you could, you could interchange them. And then we started to do a Bayesian parameter estimation approach to see the distribution, how it was shaped, and especially the two-dimensional distribution of the diffusion coefficient and this other p parameter here. And then to our surprise, but it confirmed the empirical trial and error approach. It showed a kind of hockey stick localization of the parameter. It's not well localized anywhere in this slopes or valley, you could say. The values are equally well. And so you can interchange them. So that was something completely unexpected. And this was also doing quite a challenge to get this running, because doing a Bayesian statistical estimation of the parameters in such a time-dependent model with partial differential equations was really computationally highly challenging. So we're still working on that to get it further fine-tuned. But you can get very interesting new information. But we also used our ultimate model on other species like ginseng, and that worked well. So you can also go to other plants and get different values. But now the most interesting part is what happens to mutants, and that appeared early this year in a paper. So recall that these transport values, these velocity values, they could be very well determined. So that was the thing, okay, we want to compare these mutants on this value that we can determine. We analyzed 19 batches of plants of different genotype, many individual stem segments per batch, and then in total some 157 samples were fitted each individually, and they could be fitted well. And yeah, what you see is then something like this. So it doesn't really matter what precisely these batches are. I come to that later. And we did then a statistical analysis on that, yeah, surrounding the question, the hypothesis, do all these plants have the same velocity, mean velocity? And that turned out no way. And we could identify, first doing a one-way ANOVA test, but then took a post-hoc analysis, finding some idea of what are the clusters in there. Let's see. It doesn't match up now from my laptop and this one. Oh, yeah, there it is. Yeah. So we can identify 4 clusters in this data with similar velocity. These are natural, the wild type plants, and then the low category, the low class, the 2 low clusters, they consist really of pin1 mutants, the ones lacking the green transporters. So that's one. Okay, that's nice. So first conclusion, yeah, we'll look at what happened there. Now we had this Mitchell-Kramer model with these cell files, and you had this yeah, parameter P that determined the polarity in the system. And the polarity determines, these 2 coefficients determine the velocity at the macroscopic level through this formula. Now, the presence of pins on this increases or decreases the p-value. So no pins there would be p equals 0. But p equals 0 is v equals 0, and that's obviously not the case. So you can draw a first conclusion from that, is that there must be some other protein stating involved for taking over its pin. And that was this is something that was not expected in this. And then the last 10 minutes, I want to get to how we then use the multi-scale modeling to see what happened there inside. Now, using the model. And we recall we had this stem segment high-level model that we had, the ultimate model that we got. And we now, in this model, there are 3 important parameters. There's the effective velocity in the system. That is higher, it was highly accurate, well measurable. And there are 2, A and B, and these are exchange parameters. They determine how much oxygen leaks from the central transport system to outside tissue. And we had the Mitchell-Kramer model. And in the Mitchell-Kramer model, we had the 2 important parameters, the P and the Q that appeared there. And we had the relation to the high-level macroscopic velocity. And now you can start selectively drilling down to deeper levels. So not going from bottom up, but top down. So we got to what happens actually now inside this between the 2 cells, so that we can describe the P and Q in terms of the lower level physiology of. Now, in here there are the permeability coefficients of the membrane in various directions, the betas, and what you can do, making a submodel, you can express the P and the Q in terms of these beta. And then also it turns out, so the A and the B are related to leak here on this, and they are related also to these betas, and the other parameters in here are anatomical parameters. And then we want to know what these betas are. You need to drill even deeper to what happens at this barrier. Now, on this plasma membrane, it's a charged. So it's charged. So there's a potential difference there, electric field. On either side there is oxygen, and oxygen was a weak acid that it comes in its two forms, and the balance is determined by the acidity and the acidity constant. And then there is selective exchange through the membrane, and that works for various parts differently. Protonated form can get through the membrane by diffusion. That was also part of the chemiosmotic theory. And you have a simple kind of diffusion type expression for the flux. If you go to the anion form, there can be two versions. You can have co-transport, which is essentially electrically neutral, and then it's insensitive to the electric field. And then if you work out details of how this transport can look like, you get this kind of expression and dependence on the acidity. And what is interesting to realize, so these influx proteins are this type, and because of assuming a quick equilibration here, you can actually replace this expression by something that is very similar to the protonated form. So you have effectively a protonated form transfer. And then, you have the other ones, the poorly anion transport. It will be affected by the electric field, and that is those are the green ones, the pin proteins. They form the other part, and then you can bring them together in one awfully looking expression. And if we go back to our sheet that we now have 3 new parameters: the effective apparent permeability for the protonated form and for the anion form on various sides of the cell. And then you can link all the stuff together. And then you have a full sheet of actually what you do is bridging from the lower level to the higher level, from what happens at the molecular scale to what you can measure. And there are various reasons to ignore this one. So actually we have 2 free parameters now at the bottom. And this whole complex expression was remarkably simple if you plot it. So this is the type of shape for where this side is the more of the green ones you have, the higher, and here the more of the red ones you have, the higher you get. And then recall that you can fit the velocity very well to the data. So here we have a data point for a velocity of a wild type, and the one for pin 1. So actually what you see, we determine a velocity, and you get a level set here in red and in blue. Yeah, so that and then you start getting into the statistical analysis of the whole thing. This velocity that you start with is determined up to a particular accuracy, and we took a 5% accuracy. And if you look at this, so we said, well, the fit is good when you are at this 6% or under this 6% level. So within 5%, good. And what you see here in gray is all the parameter combination that yields the same velocity within this 5% margin. And so anywhere in here you could be with these other parameters. And then we have also the A and the B parameters, these exchange parameters. These could also be fitted. And the nice thing is they only depend on the ratio only depends on this parameter, the abundance of the red ones. And so you can single out the red and the blue region for the wild type plants in red and for the pins in blue, in where you suspect and where the abundance of the greens should be. And to our amazement, what we found first of all was that if you consider pin mutants, then the abundance of red dropped, but there's no immediate relationship between this permeability constant and the abundance of red proteins there, because you knock out these and these are something else. And then we found that actually the abundance of the green ones, which were related to these pin ones, remained the same. So this left a complete puzzle. It was completely opposite than what I expected. And then you can come up with two professional conclusions that actually the role of pin 1 in this is not as presented by the chemiosmotic theory. Or what could happen is that their function is taken over by another protein and that they compensate somehow. And that will need some further molecular biological research whether you can see this expression levels changing. And we did studying expression levels is not the same as abundance levels. But we did a check on the expression levels, and what we saw indeed is this drop coincided really with a drop in the expression of these AUX-LAX proteins that are related to that. So you saw that happening, but it's not clear what can compensate it for the others. So we got to some main conclusions over the whole picture that we did. One striking thing is that we found that actually we're so focused often on a good fit, and then things are fine. But it can also be that the fit is fine, but that the biological interpretability is wrong or is lacking, and then the fit is also not good of your model. The other first conclusion, more towards the biology, that came from the statistical analysis of the mutants in view of the model, was that the pin1 proteins cannot be the sole ones responsible for this increased basal transport of oxygen in these cells, like the green one, the effect of the green ones. And the other biological conclusion that we could draw is that actually that this reduction of the permeability of the protonated form in pin1 mutants was difficult to explain, but turned out to be correlated with a decrease in AUX-LAX, the red ones expression. So that fits well. Another thing that came out of the statistical analysis is that some parameter correlations may not be found by non-dimensionalization. So they're a kind of thing intrinsic to the system in a particular parameter regime. And so, be aware then in such cases that there are parameter identification issues and also things that you would not expect. And then I would like to conclude with acknowledgement of the collaborators from biology, without whom it was not possible to do this over the years, and we happily work together and still do in different aspects.
Thank you very much. I open the floor to questions.
Thank you very much. Very, very interesting work. So when you use the Bayesian fitting, what data do you use then? Are you using sort of the overall transport data, or are you using the segments as well?
No, all data. So
All data.
so you then run into the problem of how to combine the data first of all, how you weigh the contributions of the same. and there you have some flexibility. But yeah, in this case, because it's different type of data, in dimension-wise, we used relative measures of quality for each, and then simply added the percentages, so to say, and then yeah, we were able to derive a suitable expression for the posterior distribution, and then get to the sample result.
Thank you.
Yeah, thanks for the brief explanation. I'd like to take away a couple of those examples really. I'm working at the etiology and we often forget that actually the parameters that govern certain mechanisms that you're very sure about the model is capturing actually capturing that. But I was wondering about pin 1 mutants actually. So does that confer any obvious kind of phenotypic?
Yeah, so they have a very peculiar phenotype. Yeah, so that is also where the name comes from, pin-like.
Okay.
Because they are kind of needle-like plants.
Yeah.
So they're phenotypically completely different, and
so would you expect that to be, once you're not in one, how would you expect actually from an alternative transport mechanism? What the biologists think?
no, they were quite surprised. Yeah. So through the screening that we did, the suggestion became apparent that what could take over, but it really requires a well thought experimental setup to prove that in something.
Hi. So if I understand correctly, your upscaling analysis is called 1D. And then you showed us this cross-section of the stems where you had nice 2D geometry that looks a bit like Voronoi diagrams.
Yeah.
Do you ever expect, if you were to be able to do this homogenization in 2D with some, would you expect anything to be different, or is there one main picture?
No, not. Good question. So there has been publication out where mid 2000 somehow that the more detailed cellular tissue was then upscaled to actually a 2D advection diffusion equation. We refrained from that and took a different approach because we are not sure what part of these vascular bundles are really the polarly auxin transporting cells and which are their neighbors. And so you need to upscale various versions and see what is the effect of that. But we felt that we lacked the detailed information at the moment to do that. That is something you would really want to do if you have a better insight and then see how they match together. Yeah.
And something related to it, you said that you can act on biomolecule level to have influence that you end up seeing a different plant effect.
Yeah.
Would you see a different structure of this 2D geometry?
Yeah, slightly. Yeah. So we also made microscopy images of the cross sections of pin 1 and wild type, for instance, and then you can compare the anatomical quantifiers, and you see indeed a shift, and that is then also incorporated in some of the anatomical parameters in the model. So you see differences, and in the current model, the anatomical differences are already encompassed by changing variable from so for each segment from observed correctly.
Any other questions? Last chance. If not, then let's make the speaker again. And that concludes the seminar. We are somewhat likely to meet again in 2 weeks, but details to be confirmed. If not, that might be our last seminar for this time, and we'll be starting mid-January, but I will send a message by the end of this week what happens in 2 weeks' time. Thank you.
You will stop.
Yes.
Yeah.
Yeah.